Hi, everyone. This is the February edition of Micro Seminar. My kids are home because it's rodeos in town and they get off for school for the rodeo, and so they'll be running around in the background, so don't worry about that. Today's seminar is from Dr. Kevin Kohl. Kevin just started his lab in Pittsburgh in June, University of Pittsburgh, working on all sorts of microbiome questions and how microbiomes change and help the host adapt to different environments. Before that, Kevin was a postdoc with Seth Bordenstein. And before that, he worked in Argentina with a bunch of awesome people. And then for his PhD, he went to the University of Utah and worked in Denise Deering's lab. And so without further ado, I leave it up to Kevin.
All right. Thank you. Thank you for those of you that are watching. So when I first volunteered for this, I thought I might have some preliminary data, but that's not the case. So you guys will be hearing about my PhD work and then also some of my postdoc work that I conducted in Argentina on lizards. So the title of my talk is Friends for Life, the Role of Gut Microbes in Host Physiology, Ecology, and Evolution. There we go. So it seems like in every issue of Science or Nature, there's a new paper discussing the role that gut microbes play in human health and well-being. And I think these papers really demonstrate that each and every one of us contain a significant microbial portion to ourselves. And this idea isn't too new, and I think this idea is really highlighted by a study that set out to sequence the human intestinal metagenome. And so this study found that the human intestinal metagenome contains 3.3 million non-redundant genes. And if we take the size of this circle to represent 3.3 million genes, we can place on top of that the size of the human genome, which is only 23,000 genes. And so I think this also demonstrates that our guts contain a lot more genetic and likely metabolic diversity than we contain within our own genome. And so this has given rise to the idea of the holobiont, or the idea that animals or humans don't act in isolation, but rather act as a collection of their own genome as well as the genomes of their microbial partners. And this isn't that new of an idea. We've known for decades that animals like cows are unable to degrade cellulose, and so instead they house fermenting microbes to help them with that process. And so for my research, I'm really interested in how microbial communities impact animal physiology, ecology, and evolution. And specifically, and so I set out to address some of these questions in a recent paper studying how, or presenting how host microbe symbioses could be integrated into comparative physiology. And so if we think about a microbiome, we can think about how it's influencing animals at a variety of levels of biological organization. So first we could think about how microbes influence animals at the molecular level, such as how microbes alter the metabolome of an animal in the blood or urine. We could also think about this at the cellular level. For example, how do microbial populations alter the gene expression or proteome of particular cellular populations? Scaling up a bit larger, we could think about how microbes alter organ function, specifically the performance of particular tissues or organ systems. And then I truly consider myself an organismal biologist. And so I'm really interested in how microbes can influence whole animal performance. So this could be things like digestibility or the ability to fight off an infection. And then last, scaling even larger, we could think about how microbial populations influence ecology and evolution. So for example, how do microbes alter ecological interactions? This could be things like plant-herbivore interactions. There have also been studies demonstrating that the microbiome can influence whether hosts act as vectors or reservoirs for diseases, and so that could influence disease ecology. And so with my research approach, I try to address these sort of microbe-animal interactions at these various levels of biological organization. And so specifically, I'm really interested in aspects of feeding and nutrition. So it's through these processes that animals acquire the energy and nutrients that they need to survive and reproduce. And so if gut microbes can play a role in affecting an animal's feeding or nutrition, they could play a very large role in determining an animal's fitness. And so today, I'll go through two experimental systems that I've used to sort of address some of these questions. The first is my PhD work looking at how gut microbes help herbivorous animals cope with toxic diets. And then I'll finish up with a little bit of work looking at physiological and microbial adjustments that facilitate herbivory in lizards. So we'll start with this first question. So herbivory is a common and derived feeding strategy among mammals. It's common in the fact that about 40% of mammalian species are considered herbivorous. And if we look at the pictures here, we can see that this feeding strategy has evolved numerous times independently. Now, this might lead you to believe that this is a relatively easy feeding strategy. However, that's not the case, as plants are extremely low in protein and high in indigestible materials such as fiber. And so herbivores have a number of physiological adaptations to overcome those nutritive challenges, as well as microbial partners that help them. But in addition to the nutritive challenges posed by plants, they also produce a wide array of plant secondary compounds that act to deter feeding by herbivores. And there have been decades of research investigating the interactions between mammalian herbivores and these plant toxins. And research in this area is important for a number of reasons. First of all, mammalian herbivores can play very large roles in determining whole ecosystem structure by consuming different plant species. And often their diet selection is dictated by these toxins. And so if herbivores prefer or avoid a certain plant, that can sort of scale up to determine a whole ecosystem structure. Additionally, in grazing ecosystems, there are many agricultural herbivores that are faced with these same toxins. And so by understanding how wild herbivores cope with these plant toxins, we may gain better insights into agricultural practices. And then last, many of these plant toxins actually share structural similarity to many pharmaceuticals. And some pharmaceuticals are actually plant secondary compounds. And so understanding how these toxins are metabolized could give us insight into sort of pharmaceutical metabolism. And so in 1974, renowned ecologists Freeland and Jansen published a seminal paper discussing strategies that mammalian herbivores use to overcome these toxic challenges. And since this publication, there have been decades of research investigating a number of hypotheses that they originally set up in this paper. However, one area of research, the idea of interactions between gut microbes and dietary toxins, had received very little attention since this paper. And this probably stemmed from the fact that most microbes were unable to be cultured. But with advances in DNA sequencing technology and sort of the excitement of the microbiome field, I think we can start to address some of these questions. And so for my PhD, I was studying interactions between gut microbes and dietary toxins using Woodrats as a study system. So Woodrats are a really interesting and fun study system to work in. There are about 20 to 25 species, and they tend to specialize on very toxic plants. And different species will specialize on plants with distinct chemical defense profiles. And so there's some really nice comparative work that can be done here. I've worked on a number of woodrat species, but today I'm going to focus on just a single one, the desert woodrat, and the toxic shrub that it consumes, which is creosote bush. So in the desert Southwest, there are populations of the desert woodrat that specialize on creosote bush. It can make up to 75% of their diet. Now, creosote is a really nasty plant. It covers its leaves in a phenolic-rich resin that can make up to 20% of the dry matter of the plant. This resin contains over 300 compounds, primarily phenolics, although about 40% of the resin is made up of a single compound, nor-dihydroguaretic acid, or NDGA. This compound is known to cause kidney cysts and liver damage in lab rats, and remarkably, desert wood rats consume the lethal dose of this compound as measured in lab rats every single day. And so there's really something remarkable about these animals' ability to consume these toxins. Now, there's also a really interesting sort of natural experiment in this system. So the map on the left shows the state of Utah, and the orange shading shows the range of the desert wood rat. Now, creosote bush naturally invaded from South America about 17,000 years ago, but it only came into the southern portion of the range of the desert wood rat. And so that creates this natural scenario where we have these southern populations, which I'll call the experienced populations, that have 17,000 years of ecological and evolutionary experience with creosote and its toxins. However, then we have these northern populations, which I'll call the naive populations, that have never seen creosote before. And interestingly, when we bring individuals from these populations into the lab, they exhibit differential tolerance, such that the experienced population is able to consume about 25% more creosote resin in the lab, suggestive of some sort of adaptation in this group. Now, wood rats are also really interesting from a microbial perspective due to some aspects of their gut anatomy. So the diagram here shows sort of a picture or drawing of the wood rat gut. So chamber 4 is the cecum. That's where they're doing a lot of their fiber fermentation. But what's really interesting is their stomach. So they have this sort of semi-segmented stomach morphology. And it had actually been hypothesized for over a century that rodents with this morphology might house a foregut microbial community. But this had never been tested. And so we conducted flow cytometry to measure the number of microbial cells per gram of contents in these various regions. And indeed, we found that the foregut region does contain a dense microbial community that actually has density on par with that of other well-known microbial chambers, such as the cecum or the large intestine. Now, the food material doesn't reside in this foregut chamber for very long, only about an hour and a half, which isn't really enough time for extensive fiber fermentation. And so rather, we think that this chamber might be a detoxification chamber in order to allow for these toxins to be metabolized early on in the gut before they would be absorbed in the small intestine. And this is actually a hypothesis for the original evolution of the cow rumen, that it first evolved for detoxification and then was later used for fermentation. So I'll walk through a few experiments in this system, first asking whether gut microbes facilitate the ingestion of toxins, and then also testing whether toxin tolerance could be transferred across species. So we'll start with this first one. So the first way that we addressed this question was by using antibiotic treatments to reduce the microbiota. And so here we started with a group of animals from the experienced population. So they have this 17,000 years of experience with creosote. We brought them into the lab and put them on a number of different treatments. So we had a control treatment, which are animals that are just being fed rabbit chow, and then a creosote treatment where we can add 2% of extracted creosote resin to the diet. So we can extract the creosote resin from the plant and add it to our base diet. And so the only differences are the addition of toxins, not any nutritional differences between our diets. Then a subset of animals within each of these groups were given antibiotics. We use neomycin, which is a relatively broad spectrum antibiotic, and it doesn't cross the gut lining very well. And so any differences we see should be due to reduction in the microbiota and not any systemic interactions. The animals were maintained on these treatments for about 2 weeks, and we monitored various physiological parameters such as food intake and body mass. And when animals lost more than 10% of their body mass, they were removed from the experiment. So our animals actually prefer to starve themselves rather than poison themselves. And so this body mass metric is a really nice measurement for sort of tolerance to the toxins. So I'll present the results in the form of a Kaplan-Meier survival curve. So here we have days of the experiment on the x-axis and the proportion of Woodrats remaining in the trial on the y-axis. I'll also say that this, down in the corner, I have a little diagram showing that this is from the experienced population, because I'll sort of be jumping back and forth between my two populations. So I'll always have that little figure in the corner. So again, this experiment was conducted on experienced Woodrats. And so this black line shows animals that were not given any antibiotics and were fed either the creosote or the control diet. And we see that all of these animals persist in the trial for the full 2 weeks. This blue line shows animals that were given antibiotics and were fed the control diet. And we saw that a few animals dropped out of the experiment, but this line was not statistically significant from the black line. What was most remarkable is when we look at animals that were given antibiotics and fed the creosote diet. So here we saw that all of the animals drop out of the experiment within the 2-week period. And this p-value compares the red line to the blue line, as well as corrections for multiple comparisons. So I think this experiment really demonstrates that reducing the microbiota significantly impairs these animals' ability to ingest plant toxins. I should also say that in this experiment, we only used a 2% resin diet, which even the naive animals can consume. And so treating these animals with antibiotics really reduced their ability to cope with these plant toxins, since in the wild, creosote can be up to 20% creosote resin. So then we were curious whether toxin tolerance could be transferred into the naive hosts. So again, we have this interesting natural scenario with these populations that exhibit differential tolerance to creosote resin. And so essentially what we wanted to do was transfer the microbiota from the experienced population into the naive population and see whether we could confer this ability. And so for this experiment, we started with 2 groups of animals that were collected from the naive population. For the first group, we collected donors that were also collected from the naive population. We collected feces and ground it up and mixed it into the diet of our recipients in order to create a control transplant group. For another group, we collected individuals from the experienced population, collected their feces and ground them up, mixed it into the diet of our recipients in order to create an experimental transplant group. And so here, both groups of animals were collected from the same population. The only difference is the type of microbiota that we introduced into them. So we first wanted to make sure that our microbial transplants were successful. And so we collected feces from our donors and recipients and conducted microbial inventories by sequencing the 16S rRNA gene. So this is a principal coordinates plot, which is a common way to present microbial community data. And essentially what we're looking for here is clustering. And so here we can see that our naive donors and our experienced donors cluster significantly, and so they have distinct microbial communities. Now, when we look at our recipients, we see that the control transplant animals cluster with the naive donors, while the experimental transplant animals cluster with the experienced donors. And again, the green circles are naive animals. And so before this experiment, they would have clustered with all of these purple points. But through the transplant, we effectively shifted them in this space. And so it seems like our microbial transplant was indeed successful. And so now we have these 2 groups of animals where the hosts themselves were collected from the naive population, but they're housing these different microbial communities. So then we conducted a feeding trial where we increased the concentration of creosote resin in the diet every 3 days. And then again, monitored various physiological parameters such as food intake and body mass, and removed animals from the trial when they lost more than 10% of their original body mass. So I'll first present the results for change in body mass. So here we have days of the experiment on the x-axis and percent change in body mass on the y. The boxes above the x-axis correspond to the percent of creosote resin in the diet at that time, and also when the microbial transplant was occurring. So early on in the experiment, we didn't observe any differences between our two groups. But as we transitioned these animals onto diets containing creosote resin, we observed a significant difference, such that the experimental transplant animals were able to maintain higher body mass on these toxic diets. Now, interestingly, we didn't observe any differences in food intake or digestibility. So our two groups of animals were eating the same amount of food and digesting it just as efficiently, but we observed this difference in body mass. And so this led us to wonder what the underlying mechanism was for this difference. And we thought that it could be the energetic costs associated with detoxification. So when toxins are absorbed into the body, they have to be metabolized by the liver, and this can be a very energetically expensive process that can actually be up to 45% of the basal metabolic rate of some herbivores. And so we thought that this energetic cost could be explaining the change in body mass, or the difference in body mass that we observed. And so before I get into the results supporting this idea, I just want to go through the detoxification process in order to set up some of my predicted results. So in this experiment, we're thinking about two main sites of detoxification, the gut and the liver. And so in our control transplant animals, they would be consuming a dose of toxins. So here each triangle is a toxin molecule. And in our control transplant group, we don't predict that many of these would be metabolized in the gut. And so those molecules are going to be absorbed into the bloodstream, where they then will be acted on by the liver and excreted in the urine in the form of toxin metabolites. Then when we think about our experimental transplant group, again, there's no difference in food intake, and so they're consuming the same amount of toxins. But we predict that some of these may be metabolized by the gut microbiota. And so it could be that fewer toxins are absorbed into the bloodstream. And some of these could be in the form of unique microbial metabolites. These compounds would then be acted on by the liver and excreted in the urine, again in the form of toxin metabolites. And so overall, we predict the experimental transplant group to have fewer toxin metabolites and also different toxin metabolites compared to the control transplant group. And I actually had visual evidence for this quite early in the experiment. So this picture is woodrat urine. Woodrats are desert animals, and so this is normal urine for them. They produce a very concentrated urine. And this picture actually represents the most exciting day of my PhD. So this urine was collected early on in the experiment, just when they were feeding on a 1% resin diet, and I wasn't sure really whether the experiment was working or not. But my two groups were producing visually distinctive urine. So the control transplant group was producing this sort of orange-colored urine. And this is common when the woodrats are being fed creosote resin, as they excrete these toxin metabolites in the urine. But then on the other hand, the experimental transplant group was producing this more brown-colored urine. And this is what their urine looks like when they're feeding even on a controlled diet. And so it seems like there was some sort of difference in the detoxification routes for these animals. We followed up on this by measuring the pH of the urine. So when toxins are absorbed, a very common detoxification pathway is to add glucuronic acid onto them in order to make the metabolites more soluble and easier to excrete in the urine. And so this is a host detoxification process, and it results in more acidic urine. And so on this graph, we can see the amount of urine produced and then the urine pH on the y-axis. And the control transplant group produced a significantly more acidic urine, which is indicative of more reliance on host detoxification. We also followed up on this by conducting metabolomic analysis. So this is a chromatogram of Woodrat urine, where each peak represents an individual metabolite. And we can use the area under the curve of those peaks as sort of a proxy for their concentration in the urine. And so by doing this, we identified a number of unknown compounds that were differentially produced between our two groups. I'm just showing two of them here. So these metabolites are not produced on a control diet, but are differentially produced on a 1% resin diet, such that unknown compound 1 is only produced by the experimental transplant group, and unknown compound 2 is only produced by the control transplant group. Now, creosote resin contains over 300 compounds, each of which could be metabolized into a number of metabolites. And so tracking down the chemical structure of these compounds was beyond the scope of our project. But I do think these results demonstrate that these microbial populations significantly change the detoxification routes of these animals. And then we also have some new data that we're still working on, testing whether these microbial populations can influence host detoxification or the costs associated with that. So we reconducted this transplant experiment and collected animals when they were feeding on this 2% resin diet, when we see this difference in body mass, but no difference in food intake or digestibility. And we also collected animals that were feeding on a control diet. And we collected liver tissue and conducted RNA-seq to look at levels of gene expression. And so the results that I'll show right now are looking for genes that are upregulated by creosote compared to the control diet in these two groups. And then we're looking for genes that are uniquely upregulated in each of these groups, or genes that are upregulated by creosote in both of these groups. And so here I have a Venn diagram showing the numbers of genes that are upregulated by creosote in each of these groups. So in the center, we have a list of 19 genes that are upregulated by creosote in both of our groups. And so we can think about this as a core response to creosote resin. And so this list contains 10 cytochrome P450 genes. These are well-known detoxification genes that metabolize a number of toxic compounds, and also a thioredoxin, which plays a role in oxidative stress. Now, in the control transplant group, we observed 42 genes that were uniquely upregulated in this group. So this list also contained a number of detoxification genes. We had 13 additional cytochrome P450 genes, and then 2 aldoketo reductases, and two glutathione S transferases, which are other detoxification enzymes. When we look at our experimental transplant group, here we only see 5 genes that are uniquely upregulated in this group. And we didn't see a very large detoxification signature. There was a retinol dehydrogenase, which can play some detoxification roles, but also has nutritional functions. But I think overall, these results really demonstrate that the experienced microbiota attenuates the liver's response to creosote resin. And so it's likely that the control transplant groups have to rely more on host detoxification, whereas the experimental transplant group is able to rely on microbial detoxification. And then last, I'll just present sort of our end-term metric on toxin tolerance, which is removing animals from the trial when they lose more than 10% of their original body mass. And so here we have days of the experiment on the x-axis, and the percent of animals remaining in the experiment on the y-axis. And the percentages correspond to the percent of creosote resin in the diet at that time. And so we're early on in the experiment, all animals are retained in the trial, but as we push them to higher doses of resin, we observe a significant difference such that the experimental transplant animals are retained in the trial significantly longer and are able to consume higher doses of creosote resin. And so I think these results really demonstrate that the experienced microbiota significantly improves these animals' ability to tolerate toxins. And so in summary for this portion, I think the antibiotic experiment shows that reduction of the microbiota significantly impairs these animals' toxin tolerance. And then the experienced microbiota, transferring that into the naive population, significantly increases animal performance on these toxic diets. So then I'll move into the second question, testing whether toxin tolerance could be transferred across species. So this is a project that was largely done by an undergraduate that I mentored in the lab, Ashley Stengel. And I think this idea of transferring across species is really important for the idea of whether we can use what we're learning in wild herbivores in agricultural herbivores. And so for Ashley's project, we focused on a specific class of toxin and specific type of bacteria, which are tannin-degrading bacteria. So tannins are a specific class of chemical defense, and they represent nutritive challenges because they bind to dietary protein and also to digestive enzymes and prevent animals from digesting and absorbing nutrients. And so they represent a really big nutritive challenge to herbivores. And because of that, they're of really large concern in the agricultural industry. And so there's been interest in isolating tannin-degrading bacteria. And indeed, scientists have isolated tannin-degrading bacteria from a number of animal hosts. So the first tannin-degrading bacteria were isolated from the guts of koalas in the year 1990. And since then, scientists have isolated tannin-degrading bacteria from a number of other mammalian hosts. However, what we still didn't know at the time was whether tannin-degrading bacteria are actually functionally significant to their hosts. That is, we didn't know whether hosting tannin-degrading bacteria actually allowed herbivores to consume higher doses of tannins. And so we set up an experiment to address this knowledge gap. And so the first thing we did was to isolate tannin-degrading bacteria from the woodrat gut. So this is an agar plate and that's been overlaid with tannic acid. And the tannic acid binds to the proteins and precipitates them out, creating this opaque surface. And then we can culture fecal material on this plate, and we'll get some of these colonies that create zones of clearance. And that's where they are dissociating these tannin protein complexes. And so using this technique, we isolated 3 species of tannin-degrading bacteria from the woodrat gut: Bacillus subtilis, E. coli, and Enterococcus fecalis. And so then we set up an experiment where we conducted several transplants from the woodrats into lab rats. Now, woodrats and lab rats are in different taxonomic families and have been separated for about 25 million years. And so while they might look somewhat similar, they do represent a substantial host jump for the microbiota. So in the first group, we collected feces from woodrats and autoclaved them in order to sterilize them and inoculated or mixed it up into the diet of our recipients in order to create a controlled transplant group. In the second group, we cultured these tannin-degrading bacteria and then grew up an overnight culture. We did a mixture of these 3 species and introduced it into the lab rats in order to create an isolates transplant group. And then last, we collected whole woodrat feces, ground it up, and mixed it into the diet in order to create a community transplant group. Similar to the previous experiment, we conducted 16S rRNA inventories and demonstrated that these microbial transplants were indeed successful. And so here we have these 3 groups of animals where the hosts themselves are probably genetically identical, but they differ in the type of microbiota that we've introduced into them. So we then conducted a feeding trial where we increased the amount of tannic acid in the diet every 3 days, and then held them on a 12% resin diet for 9 days. We also monitored various physiological parameters, such as food intake and body mass, and then removed animals when they lost more than 10% of their original body mass. So first, I'll show the results for toxin intake. And so here we have our 3 groups and the cumulative tannic acid intake over the first week of the experiment. And we can see that both the isolates and the community transplant groups were able to consume more tannic acid, about 12% more. And so this really represents the first demonstration that these tannin-degrading bacteria are functionally significant to their hosts. Now, even though these groups were consuming higher doses of toxins, we actually observed lower indicators of liver damage. So we collected serum samples and measured the activity of an enzyme, alanine aminotransferase. So this enzyme is primarily produced in the liver, but as liver cells are damaged, the enzyme is dumped into the serum where its activity can then be measured. And so higher activity is indicative of more liver damage. And so at least for the community transplant group, we can see that even though this group is consuming higher doses of tannic acid, we have lower indicators of liver damage. And then last, we were interested in their persistence in the experiment. So again, a Kaplan-Meier survival curve looking at how animals persisted in the trial. So this blue line shows the control transplant group. So we see that all animals drop out of the trial. But when we look at our isolates and community transplant groups, we saw a significant difference such that these groups were retained in the trial significantly longer, really demonstrating that these tannin-degrading bacteria allow these animals to persist on toxic diets. And so then we were curious whether this function could be retained even in the lack of dietary toxins. And so this graph is the graph that I just showed on the previous slide. And so then the animals entered a 1-month recovery period during which they were not fed any tannic acid. And then we basically reconducted this feeding experiment without reconducting the transplant or inoculation aspect of it. And basically we found the same results, such that the isolates and community transplant groups were retained in the trial significantly longer. And I think these results are important for the idea of using these microbes in an agricultural setting, whether herbivores would have to be constantly re-inoculated with these novel microbes, or whether they might be retained even in the lack of dietary toxins. And it seems like, at least in our system, these microbes and their functionality persist at least for a 1-month period. So it seems like these tannin-degrading bacteria do allow hosts to consume more tannins. And again, I think this represents the first test of functional significance for these types of bacteria, even though scientists have been isolating them since the year 1990. And I think it also demonstrates that microbes from wild herbivores could be of use to agricultural animals. And so I think we can also take some of these results back to this paper originally published by Freeland and Jansen. And I think these results show that gut microbes do indeed facilitate the ingestion of dietary toxins. And while my work has been largely focused on woodrats, I think they do have implications for herbivores at large. So first of all, I think microbes likely drive the foraging strategies of other mammalian herbivores. There are a number of herbivores that specialize on other toxic plants. And as I said in the beginning, herbivores can play very large roles in determining whole ecosystem structure. And so I think there might exist some very interesting natural systems where a microbe alters the feeding preferences of an herbivore, which then scales up to determine whole ecosystem structure. And I think understanding those ecological cascades would be really interesting. Second, I think microbial populations could represent routes of very rapid ecological adaptation to changing environments. So with patterns of global climate change, plants are actually predicted to produce higher concentrations of plant secondary compounds. And additionally, through changes in land use practices, there are large shifts in plant communities over the landscape. And so herbivores may be faced with novel plant toxins in higher concentrations. And so rather than have to evolve new detoxification pathways in their liver through classical Darwinian evolution, they may be able to pick up a new gut microbe that conducts those processes for them. And then last, I think the Woodrat experiment really the lab rat experiment really demonstrates that wild herbivores could be a source of novel microbes for use in the agricultural industry. And like I said, there are a number of woodrat species that specialize on different plants. So we've also conducted inoculations into lab rats from another species that specializes on cactus, which is rich in oxalate, and sort of found similar results. And we also have some data demonstrating that the microbiome is important for facilitating tolerance to terpenes, another class of chemical defense. And so I think wild herbivores could be a source of these novel microbes. So to finish up, I'll just talk briefly about some of my postdoc work, testing whether physiological and microbial adjustments facilitate herbivory in lizards. So I spent most of this time talking about mammalian herbivores, where herbivory is the most common feeding strategy. Again, 40% of mammals are considered herbivorous. But if we look at reptiles, herbivory is actually very scarce. Actually, only 3% of lizards are considered to be herbivorous. And so for decades, ecologists and evolutionary biologists have theorized about what limits herbivory in squamate reptiles. And so one idea is that it could be limited by ecological context, that herbivory isn't the best feeding strategy, and so it only evolves in areas with low prey availability, so places like arid environments or islands. On the other hand, people have theorized that lizards may have intrinsic limitations to an herbivorous lifestyle. So this could be things like poor chewing efficiency, low digestive efficiency, or perhaps they lack the appropriate gut microbes. And so I set up a feeding trial to sort of test this side of the question. And so for this, I went down to Argentina to study the family Leolemidae, which is a really interesting family of lizards, where herbivory has actually evolved 18 times independently. And if we look across all other lizard groups, herbivory has only evolved 11 times. And so there's definitely something special about this group in its ability to evolve this feeding strategy. And previous work had demonstrated some physiological adaptations in this group. So O'Grady et al. Went out and collected individuals from the wild and measured their snout vent length, which is sort of their body length, and also their small intestine length, and found that herbivorous lizards had longer small intestines that were actually about 70% longer than omnivores or insectivores. And a longer gut sort of facilitates increased interaction between the animal and the food material and helps to increase their ability to digest it. Now, what we don't know from this experiment is whether these are truly adaptations, evolved adaptations, or perhaps phenotypic plasticity, since all of these animals were collected from the wild, and so they were all feeding on their natural diets. And so they don't really test the limits of herbivory. And so I conducted a feeding trial that aimed to address these questions. So first, can an omnivorous lizard digest and maintain nitrogen balance on a plant-rich diet? Second, does a plant-rich diet induce physiological adjustments? And then last, does a plant-rich diet induce microbial adjustments? And so I conducted this experiment on the species Leolemus ruibali, which is a small omnivorous lizard that lives in the foothills of west central Argentina. And so in the wild, it only incorporates about 16% plant material into its diet. And so what I did is I brought individuals of this species into the lab and fed them either a mixed diet of 50% rabbit chow and 50% ground mealworms, or a plant-rich diet of 90% rabbit chow and 10% ground mealworms. So we measured food intake for 40 days and also collected feces and urine pellets in order to measure digestibility of dry matter and digestibility of fiber, and then also nitrogen balance, which is the amount of nitrogen going into the animal minus the amount of nitrogen coming out of the animal in urine and feces. And then at the end of the experiment, we dissected the animals to look at aspects of gut anatomy, and then also conducted microbial inventories in various chambers. So I'll just go through some first performance parameters for the animals. So food intake did not differ between my two groups. Body mass also didn't differ between my two groups, and there was no significant change over the experiment. The dry matter digestibility was lower in the plant-rich group. And this is because of the fiber material that they were consuming that was also showing up in the feces. So just bulk dry matter digestibility was lower in this group. And so then we were interested in nitrogen balance. So again, this is the amount of nitrogen going into the animal minus the amount going out. And so animals want to be in positive nitrogen balance. And so this is a very common way to present nitrogen balance data. So on the x-axis, we have nitrogen intake, and then on the y-axis, we have nitrogen balance. And so essentially, animals want to be above this 0 line. And for the most part, we see that animals in both groups were in positive nitrogen balance. We have 2 of these plant-rich individuals that were in negative nitrogen balance. This one that's very negative was actually the smallest animal in my experiment and had the highest nitrogen content in its urine pellets. It didn't exhibit any obvious signs of poor health, but some of those factors, its low body mass and high urine nitrogen, sort of pushed it into negative nitrogen balance. But overall, it seems like this naturally omnivorous lizard is able to maintain positive nitrogen balance, even on a plant-rich diet. We were then interested in aspects of gut anatomy. And so similar to the O'Grady et al. Paper, we measured snout vent length or body length and small intestine length. And we found that individuals fed the plant-rich diet had significantly longer small intestines for a given body size. Now, this change was only a 17% difference, whereas O'Grady et al. Observed a 70% difference between her groups. But it does seem that this plant-rich diet is able to induce changes in gut anatomy, perhaps facilitating the better digestion of this diet. And then last, we were interested in aspects of microbial diversity. So we inventoried microbial communities of various gut regions, and this will just show the results for FAITH's phylogenetic diversity. And we saw that lizards fed the plant-rich diet had significantly more diverse microbial communities, at least in the small intestine and the fecal communities. And these results are consistent with studies in mammalian herbivores, where herbivorous mammals have more diverse microbial communities to other feeding strategies. We were also really interested in whether there might be some microbial taxa that would correlate with whole animal performance. And specifically, I was interested in fiber digestibility. And so we ran some statistical models and that corrected for multiple comparisons. And the one that fell out as most significant was a correlation between the relative abundance of disulfovibrio in the small intestine and the animal's ability to digest fiber material. So animals that had higher abundance of disulfovibrio also exhibited higher fiber digestibility. Now, disulfovibrio is a sulfate-reducing bacteria, and so it could be playing a role in hydrogen reduction. So when herbivores consume fiber, it's broken down into short-chain fatty acids, but through this process, there's also a production of hydrogen that has to be dealt with in the system. So this schematic shows hydrogen reduction in the bovine rumen. And here, most of the hydrogen is dealt with by methanogenic archaea that converted into methane. So this is when you hear about cows burping out methane, it's through this process. Now, there had been a study on marine iguanas looking at their microbial communities, and they actually found that marine iguanas lack these methanogenic archaea. And so instead, they had they saw higher abundances of sulfate-reducing bacteria and acetogenic bacteria. And so I think my results are sort of consistent with this idea that reptilian herbivores may be relying more on these alternative hydrogen reduction pathways compared to mammalian herbivores. So in summary, just sort of going back to some of these questions, can an omnivorous lizard digest and maintain nitrogen balance on a plant-rich diet? We saw that the answer was yes for the most part. Does a plant-rich diet induce physiological adjustments? Yes, somewhat. We saw a 17% increase in small intestine length, which is not as great as the 70% difference observed by O'Grady et al. And this could be due to either that the 17% difference represents the sort of the limitations of phenotypic plasticity, which is then exaggerated over evolutionary time to yield a 70% change. An alternative explanation is that my feeding trial maybe wasn't long enough. At 40 days wasn't long enough to induce a 70% change. And then last, does a plant-rich diet induce microbial adjustments? Yes, we saw more diverse microbial communities in the plant-rich diet group, and also some nice correlations between sulfate-reducing bacteria and the ability for individuals to digest fiber material. So in conclusion, it seems like physiological and microbial limitations do not sensu strictu constrain herbivory in Leolemus ruibali. And so rather ecological contexts could be more important in driving the evolution of this feeding strategy. But I do want to point out that these lizards were living a very easy feeding lifestyle. They didn't have any costs of breeding. They were literally being force-fed, and so they didn't have any foraging costs. And so even though they were able to consume a plant-rich diet group in the lab, or a diet in the lab, they may not be able to do so out in the wild. And then last, I think the roles of certain microbial groups might differ between mammals and reptiles, especially some of these hydrogen-reducing groups. And I think that would be a really interesting area to sort of follow up on. And so with that, I think these experiments really demonstrate the roles that microbial communities can play in the ecology and evolution of holobionts. And so while we're constantly being surprised at the role that microbes play in human health and well-being, I think we're constantly going to be surprised at the roles that these microbes play in ecology and evolution. And I'm really excited to think about some of the other functions that may be brought about by these microbial communities. And so with that, I'll thank a number of collaborators and advisors through my PhD and postdoc, as well as a number of funding sources. And then I'm happy to take any questions.
All right, that was awesome, man. Thank you very much.
Yeah.
So if anybody has any questions, feel free to tweet them at Microseminar or join the Hangout, or there's a YouTube comments too. But I actually have a couple questions in case anybody, well, while we see if anybody actually has any. So my interests are actually in microbial genomics and kind of adaptation of microbial genomics. And it seems like this is a pretty awesome system to look for where the pathways are actually coming in potentially to help with things like tannin degradation. And so have you started to look at the Woodrat microbiota and then the relatives of the microbiota in the non-toxin degrading species, just to look for kind of where those pathways are coming in the microbes, or is that kind of on the docket, or what's up there?
So we haven't compared the two populations. We have done some metagenomic, well, okay, actually we've done a bit of metagenomic work in the creosote system, looking at individuals that were fed either a control diet or a creosote diet. And we see an increase in the abundance of genes associated with breaking down aromatic hydrocarbons, which is perfect because that's essentially what we're giving to them. Some of those genes seem to be coming from lactobacillus. That system is a little more challenging since the chemical diversity is just so high. And so I think some of these simpler systems are a great place to go there. So like the tannin-degrading bacteria, oxalate is another good system because it's a single compound that's only broken down by microbes. We've also done some comparative work comparing a terpene-feeding woodrat to a cactus feeding woodrat. Cactus doesn't really have a lot of these toxins. And in our juniper feeding woodrat, which has a lot of terpenes, we do see higher abundance of some of these terpene degrading genes. So yeah, we do have some evidence. It's just with these very chemically complex mixtures, it's a little hard to track down exactly what the mechanism is.
Cool. And then the other question I have too is I guess in my knowledge of the mouse and rat ecologies is kind of limited, but from what I remember with the microbiota, you see these geographic patterns in kind of human associated rat and mouse populations where there's like latitudinal gradients and stuff like that. Mm-hmm. So do you know if the same thing's happening in, like, do you see that in wood rats? Potentially, does that interact with their ability to degrade toxins at all? Or is there kind of, it seems like there could be some interesting things going on at that level. So.
Yeah, so the studies that I've conducted have only been on these 2 populations. But since I've left the lab, there's a new postdoc who has basically gone out and collected as many populations as we could get our hands on. So they've went on numerous trapping trips to California, Arizona, Nevada. And so there we're looking at the effects of natural diet, geography, species, host species, things like that. From what I understand, those have been sent out for sequencing. So those results are to come, I would say.
Cool. I'm getting feedback, so I have no clue where that's coming from, but otherwise, I don't see any other questions, and so I'm just gonna thank you one more time.
Okay.
That was awesome. It's awesome to see talks like this and get them up online. So thanks very much.
Thank you.
And if anybody has any questions, feel free to contact either Microseminar or Kevin. I've tweeted his handle out a bunch of times, so he's very responsive on Twitter. So if you're interested in any of this, get in touch with him. And once more time, thank you much, man. This is awesome.
Right. Yes, thank you.